Hello everyone, today we'll be talking about hepatitis serology, and we want to explain if somebody have an acute infection, a chronic infection, is recovering or is immunized. So the first thing is that we have to know, we have two things to deal with: the antigens and the antibodies. Okay, so for the antigens, we have surface antigens, HB core antigens, and we also have the infectivity antigen, HBe antigen. For each one of these antigens, there are antibodies that develop. For the H, that we have the anti-HBs. For anti-HBe, and for the core, we develop the IgM and later the IgG. To understand these, we'll take a look at the viral particle have a surface protein coat, then we have the core, then we have the DNA inside. So the surface antigens are here. These are the surface antigens. The core antigens are right here. Okay. To know if somebody have an acute infection, chronic HBV, if they're in the recovery period, or if they're just immunized, we will have to take a look at the antigens and the antibodies. So for the acute person, acute infection, we will find the surface antigens in the serology. And for the antibodies, we will find the IgM, the anti-core antibodies. For the chronic infection, we will still find the surface antigens because the body haven't developed the antibodies anti-HBs yet. So we will find, and we will also find the IgG anti-core antibodies. For a person who's in recovery, there is no antigens. We should be finding anti-HBs, anti-surface antigens, and also the anti-core antigens, IgG. For an immunized person, we will find anti-HBs, anti-surface only, because the immunized person have developed antibodies to the surface antigen, not to the core, but the infected person have developed antibodies to the core as well. About the HBe antigen and antibody, simply if you see the HBe antigen, that means the virus is actively replicating, which means the person is high infectivity. If you see the anti-HBe, that means the person has low infectivity. For the HB core antigen, this one we will never see in serology. For the time frame of each antigen and antibody rising and going down, we will probably have to make a different video to make this one short and simple. I hope that was helpful. Thanks for watching.